Okay, Andy, one of the things that you have to do with this microscope is you have to set up your samples or sample collection tubes before you get started. The way you do that is to put you have a tray that these small tubes are put into. They're put in kind of a situation or a position where you actually load the samples into the cap. And in doing so, we also have a liquid that we're going to drop them into in there. So as you can demonstrate how to load the sample tubes into this stage collection device.
So this is the PCR tube holder, and I'm looking to load a few of these tubes. I'm going to just happen to load 4 of them, and I'm going to then put some liquid in it. So here's over here. I'll load the software. I'm going to put here. I'm now going to get some reagents.
And how much are you loading on there? In this case, you're going to load 22 microliters?
We're loading 22, yes.
Now, different types of caps can load different ones. In fact, in our tubes, because we use different PCR tubes, these are 0.2 milliliter tubes. We can actually, our tubes, we can load as much as 50 microliters.
Right.
One of the cautions you have to have about these under-counter tubes is that they're next to the light source, and over a period of time, some of this liquid may evaporate. So you may actually have to replenish it or only put enough samples in there or tubes in there that you can immediately collect within a few minutes.
Correct. So now I'm going to, so I've loaded all 4 of these, and the machine will now bring them underneath, and now these PCR tubes are directly underneath the area where we'll be collecting the sample.
Mm-hmm. And on the screen, you can actually select what you have. Right now, you have it set to drop into space somewhere.
Right now, I have no cap, and what I'll do is that put it into, for example, this particular cap, marked SR. And what we can do is that we can go to the 40x objective, and then find a sperm right there. We can designate it like that, and cut it. Now that sample's fallen into the cup.
That's neat.
Okay.
There's one more issue when you're looking at these slides or looking at these samples, is that the slide has to be upside down.
Correct.
Because we're cutting and dropping into it. Occasionally, we've had some of our analysts put it in right side up, not thinking, and when they cut it, it doesn't drop anywhere. But so it's a good point to remember when you're putting these slides in there that you have to put the membrane side inverted so that we'll be able to be cut and drop into the top of the tube.
Absolutely.
Okay, with the slides you're looking at here, this is a pin slide. Look at bright field, and this is what you see normally. And you see, as Andy talked about, the architecture, the epithelial slides, and you can see the epithelial cells, and you can see the spermatozoa in there, but you can also see the dots or the holes in the matrix or the pin that you're looking at. One way you can look at this better is with fluorescence.
And as we said yesterday, as we said, these pen slides have a problem in the fact that there's somewhat autofluorescence with the architecture. To demonstrate that, we're going to go into fluorescence mode here by switching the microscope, bring up fluorescence, and you see how, by changing the exposure time, that we have the dots are going to get in the way of looking at the spermatozoa. We also see some autofluorescence here with the green dots, and it can interfere with the signal, Pat. So
we're still able to see the outlines of cells and the bright spots, which are some of these are spermatozoa. But again, you have that high background that's a characteristic of autofluorescence.
Right.
And that's actually the pin film itself or foil itself having autofluorescence or having some of its own fluorescence or
Correct.
emissions.
Well, we've been trying to improve this membrane. There's been other technology that's come to avail to us. And an example of that is we're going to move to another slide. This is called the director slides or the ETC slides, energy coded slides, where it has the advantage, and we'll go back to bright field here to first show you. We have now a section here. We have a piece here. We obviously see the spermatozoa here. See that, Pat?
Yes.
But you notice here that there's no background.
Mm-hmm.
And this is one of the advantages of these energy coded slides. Another advantage of these energy coded slides is that there is very little autofluorescence. So we'll switch from bright field into fluorescence, and you'll see here now that there's not as much background autofluorescence. Also, there's no architecture. The pores aren't there.
Right. This is a much easier way to see it. And the other slide, which we looked at previously, the pin slide, we saw the autofluorescence on that, but in that one, even the other fluorochromes like DAPI cause even more autofluorescence. And one of the benefits of this slide is if you go to the DAPI setting, you'll see that there's not even any autofluorescence on this slide.
So we'll go to the DAPI setting here. And so right now we're in the DAPI setting. We're in the DAPI setting now, and get a little more signal here. You can still see the sperm there, but there's virtually no background in the membrane.
Right.
We can go back to the pen membrane and show that the autofluorescence. How about that?
Okay.
We're going to go back to the pen membrane. And we can see that we're now in the pen membrane, and again the background's even higher with the DAPI excitation compared to this is the only blue excitation. Okay.
Yeah.
All right. The next part of the development was to go to using the ETC slides, the energy transfer coded slides,
or the director slides.
Director slides for fluorescence. And we have a sample of that in one of our other slides. Slide position number 1. Oops. No, it's 3. Hang on. I'm sure they'll edit that out. We'll move to. We're now moving to the director slides, and to find. Looking for stuff.
Are you capturing this also? Okay.
Here we go.
What I'm going to do is ask him when some of these things have different intensities, so we can move this back over here. And I wanted to show the strength.
Well, we can. Okay. I'm going to bring this down because it is way too high. How about this? How about like that?
That's good.
Okay. I'm going to go back to 40x. Okay.
Because in reality, the background cells should not be fluorescing, only the spermatozoa. So that way you can examine this slide. And these bright spots that you're seeing on, those are the spermatozoa. That's one of the wonderful things about checking or examining this type of slide in fluorescence, is that you're missing a lot of the background, or you're not seeing the background is blacked out, and only these bright spots. And because of that, you're able to reduce the magnification when you're searching. And every time you reduce the magnification by 50%, you're actually quadrupling the area that you can search at the same time. So these bright spots against a black background or dark background are very easy to search, very easy to find. And if you only have a few spermatozoa, it makes it really easy to find them.
Exactly. Now I'm going to switch now back to the DAPI with these director slides, and you can see how that it has very little autofluorescence in the background, and yet you can see. I think that looks like a is that an E slide?
Looks like an epithelial cell, but before I go to the issue with that, the DAPI is a generalized nuclear stain.
Right.
So you're going to stain both epithelial DNA and sperm DNA in the same field. The Alexaflora that we were using before, you saw with the green color, those are specific for spermatozoa. So this is the way that this particular product can give you you can identify epithelial cells, but you can also use the green fluor to examine spermatozoa specifically. So in that way, it gives you kind of a nice image. However, one of the problems with these new director slides is that, and if you switch to bright field, you'll see what's happening here. Here's the same image, but now we're going to bright field, and you see all these crystal formation in here. So now it becomes virtually impossible to examine this slide under bright field because of these crystal formations. This is something that can be fixed, but as we stand right now, that's one of the issues with it. To show how hard it would be to see a spermatozoa, why don't we outline one here, and then go to bright field, and see if it can be seen. You see under bright field that area is circled, and it becomes, well, it looks like there's something inside that circle.
It'd be tough. It'd be tough to find.
If you had to look at all this background and crystals and drops, it'd be very difficult to find that on there under bright field.
Right.
Those crystals that we're seeing also could present a pretty severe problem in trying to cut that sample off of there.
Correct.
They might be something that could hold it on there or catch it or prevent it from dropping into the cap.
Now, one thing I wanted to show you, Pat, was that we're going to go back to the blue excitation is an example of how we can quickly potentially retrieve these sperm off these director slides. I'm going to go to the move and cut function. Oops, I'm going to go to, what happened here? Shouldn't have done that. All right, go back to here.
I'm going to have to turn the intensity up some.
Oh, I know what happened. Okay, go here. Okay, so here we are with the sperm showing in the green. What I can do now is with these director slides, I can knock off these sperm by drawing a little circle around them. You can see the fluorescence is gone, which indicates that they're no longer on the slide. This is again different than the pen membrane slides where you would have to circle around them. So here we're actually then just with the process called lift, laser-induced forward transfer, we are simply knocking the sperm off the energy coating.
One of the things that someone looking at this might not generally appreciate about fluorescence is it's a much dimmer form of illumination. And we have our settings here at a very high rate. In other words, if you're looking at bright field, you can see that fairly easy. But this is a situation where, as I talked about earlier in the class, the objectives on the microscope are extremely important. And this is an area, if you're going to do fluorescence on your microscope, you want to get some of the best objectives that you can because the light gathering and their ability to have bright surfaces. In this case, a very inexpensive objective may work great for bright field, but it may work very, very low or hardly at all for fluorescence.
Absolutely.
And when we're talking about the chromosome paint, we're actually doing FISH, or fluorescent in situ hybridization. We see that we need as much light as we can get. So this is an area maybe on the 40x or 63x objective where you want to get some really good objectives to enhance the ability to collect the light of fluorescence.
I can't agree with you more.
Okay. Oh, the other thing that we might want to talk about, and to show that dim and light and what happens if you're trying to move your image at these low or these slow time frames, like here you have like a 1/2 second capture.
Yeah, this camera is an analog camera, and it has normally runs at 30 frames per second, but right now we're taking half seconds, and if I move the slide, you notice that it doesn't take, the image is difficult to pick up quickly, and it's hard to focus.
Yes, because you're seeing everything at a much slower rate. This is like the old scan line in the scanning electron microscopes where it would go down 2 or 3 seconds for the whole screen. And you're often having to work at anywhere from 1/2 to 1/4 second of time or image collection time to do this.
Okay. Good.